All right, let's jump right in. For most of human history, the overwhelming majority of viruses on our planet have been, well, completely invisible to us. But that's all changing thanks to a brand new field called Viromics. Seriously, it's like we've just been handed this super powerful microscope to explore a hidden universe that's just teeming with life. And what we're finding? It's rewriting the rule book.
And I want to kick things off with this quote, because it just perfectly sets the stage. When we say viruses are everywhere, we mean it. We're talking the deepest oceans, the hottest volcanic springs, the soil right under your feet, and yeah, even inside our own bodies. The planet is it's literally saturated with them. So that leads to the big question, right? If viruses are the most abundant biological things on the planet, what are they all doing? And the honest truth is, for most of them, we have absolutely no clue. It's like we're staring into this massive universe of, you could call it viral dark matter. We know it's there, we can feel its presence, but we're only just now starting to figure out what it is.
And to really get why this dark matter has been hidden for so long, you gotta look at how we used to study viruses. And honestly, it was a huge problem.
Just think about this for a second. For decades, if you wanted to study a virus, you had to catch it, right? Then you had to find its one specific host, say a certain kind of bacteria, and then you had to somehow get that host to grow in a little petri dish in the lab. If you couldn't pull off all of those steps, the virus stayed completely invisible. It's kind of like trying to understand the entire animal kingdom by only looking at the animals you can keep in a zoo. You'd missed almost everything. Okay, so how do we see the unseeable? Well, this is where the new toolkit comes in. It's a total revolution that lets us just bypass the petri dish altogether.
And here it is, the key idea, Viromics. It's a complete game changer. Instead of trying to grow the virus, we just read its genetic code directly from an environmental sample. We just skip that impossible step of cultivating it and go straight for the blueprint.
So how does it actually work? Well, it's basically a three-step process. First, you go out and collect a sample. Could be a scoop of dirt, a liter of ocean water, pretty much anything. Step 2, you sequence everything in there. All the DNA and RNA from bacteria, from fungi, and of course from the viruses. This gives you this massive jumbled mess of genetic code. And then the final step, and this is the real magic, is using these incredibly powerful computers to sift through all that data, find the little pieces that look viral, and stitch them back together into whole genomes. And the results from this new approach? They've been nothing short of staggering. I mean, we're not just adding a few new dots to the map of life. We are literally redrawing the entire map of the viral world from scratch.
Now check this out, because what's so cool here is the sheer diversity it's uncovering. It turns out about half of what we're finding are these things called caudivoricites. The classic head-tail phages that kind of look like little lunar landers. But we're also discovering huge numbers of RNA viruses, and even these so-called giant viruses that are so big they can be larger than some bacteria. This is a whole world of diversity we just had no idea was there.
And to put a number on it, just one major database now has over 5 million unique virus genomes found using these methods. Let that sink in. 5 million. That's an explosion of knowledge, and honestly, we are just getting started. And as we start cataloging all these new viruses, we're starting to see patterns. The first big one is that most viruses, they're specialists, they're homebodies. A virus that's adapted to a deep-sea hydrothermal vent is probably not going to be the same one you find in forest soil. It seems like about 95% of them really stick to one specific type of environment, most likely because that's where their hosts live. Okay, but here's the twist. While most of them stay local, the few that do travel are serious globetrotters. I mean, scientists are finding viruses that are nearly identical in samples taken from opposite ends of the globe. A virus in the Atlantic Ocean could have a twin in the Pacific. This is hinting at a globally connected virome that we're only just beginning to map out.
All right, so we've found millions of new viruses. That's huge. But the next question is even more fascinating. What can they actually do? Well, it turns out they're not just these passive little replicators. They're tiny puppet masters.
And the key to their power seems to lie in something called auxiliary metabolic genes, or AMGs for short. Now, these are extra genes that a virus carries around. They're not for making more copies of the virus itself. Oh no. They're for hijacking and reprogramming the host cell's metabolism. So think of it like this. The virus breaks into the cell, the factory, and then it pulls out its own special toolkit of AMGs to rewire the assembly line, forcing the factory to become way more efficient at pumping out new viruses.
And these AMGs give viruses some pretty incredible superpowers. For example, we're finding viruses in the ocean that carry genes for processing sulfur,
which could actually impact cloud formation and, by extension, the global climate. We're finding viruses in contaminated soil that carry genes to help their bacterial host resist things like arsenic and pesticides. And this one is amazing. We discovered these hyper-compact versions of CRISPR gene editing tools inside viruses, which could totally revolutionize biotechnology.
Now, this is all incredibly exciting. But as you know, every discovery just opens up a whole new set of questions. Viromics has shown us just how massive the viral universe is, but it's also shined a huge spotlight on the great unsolved mysteries that researchers are now desperately trying to solve.
So, mystery number 1. Who does this virus infect? This is the host problem. We can pull a brand new, never-before-seen viral genome out of a sample, but for the vast majority of them, we have absolutely no idea what organism it actually infects. It's like we've got the key, but we're standing in a hallway with millions of unmarked doors.
And mystery number 2 is the activity problem. See, just finding a viral genome doesn't tell you if it's part of an active, raging infection, or if it's just a dormant piece of DNA kind of hanging out in the environment. Is the virus alive and replicating right now, or is it basically a genetic fossil?
So this really puts the two big challenges side by side. Predicting the host? Yeah, that's like finding a key, but having no clue which of the millions of doors it opens. And figuring out if it's active? That's like seeing a car parked on the street. You can describe the car perfectly, the make, the model, the color, but you have no way of knowing if the engine is on, off, or totally busted. Solving these two puzzles is really the next great frontier for viromedics.
So when you step back and look at the big picture, Viromics has opened a book we never even knew existed, a genetic encyclopedia of our planet, and we've really only just begun to flip through the first few pages, discovering this world of unimaginable diversity and power. And it leaves us with this one really provocative thought. What revolutionary technologies, what new medicines, what new understandings of life itself are just waiting to be discovered on the very next page? The search has only just begun.